So when we say our energy demand is increasing, we are importing more quantities of crude. We are increasing our refining capacity. We are increasing the use of automobiles. So all these have culminated into generation of some pollutants. And we being in the petroleum laboratory, it's our mandate that petroleum related pollutants or this pollution have to be taken care of in a scientific manner. So our target is to develop a very stable microbial consortium comprising of bacteria, funguses, and all other things, and then that consortium can be sustained in a very a way that in which it can be handled easily into, as you mentioned, oil spills. Oil spill has been historically a very grave problem.
Welcome back to another amazing episode of the Clampanor Show. This episode is in collaboration with CSIR Indian Institute of Petroleum. In this collaboration, you would see a series of 8 episodes featuring eminent scientists from CSIR-IIP who are actively working to build an Atmanirbhar Bharat while decarbonizing India's energy footprint. On today's episode, we are speaking with Mr. Sunil and Dr. Pankaj. These scientists have actually identified a fungus which can remove toxic, carcinogenic, polycyclic aromatic hydrocarbon from the environment. But wait, identifying a fungus and then this fungus actually going on and cleaning this pollutant, how is this going to really happen? How can this fungus actually clean an oil spill? What does it take to scale such kind of technologies? Please tune in and find answers to all such questions. I hope you enjoy the conversation. While you do that, please do not forget to hit the subscribe button. Welcome Sunil ji and Pankaj ji on the Clampanor Show. Today we are basically talking about the technology that you guys have developed, Pyrene remediation by Trametes maxima, which is a fungus. So, firstly, we'd like to start with why did you develop this technology, and also please break down the words Pyrene, remediation, and Tramita maxis.
All right. A very good morning. Thank you for taking this subject into your discussion. Actually, we know that our population is increasing at a very rapid pace, and this has necessitated the speedy economic growth, and this involves our ever increasing energy demand. So when we say our energy demand is increasing, we are importing more quantities of crude. We are increasing our refining capacity. We are increasing the use of automobiles. So all these have culminated into generation of some pollutants. So these pollutants, let's say the petroleum generated pollutants, they originate from a deliberate or an unintentional or accidental causes. So these, as you asked, Pyrene and remediation, these words have genesis from these pollution sources. So when we drive an automobile, when we use automobile, when we say that our industries are operating at a very higher capacities, our refining has increased. So these all generate lot of petroleum related pollutants, and they accumulate into the environment. So the pyrene belongs to a very toxic class of hydrocarbons termed as polycyclic aromatic hydrocarbons. So these polycyclic aromatic hydrocarbons, they are abbreviated as PAHs, and they are globally of very high concern because they are highly toxic, mutagenic, and they have been taken up by the plants, and they have found route into our food chains. So due to these associated toxicity, these PAHs have been studied very widely across the globe, and we being in the petroleum laboratory, it's our mandate that petroleum related pollutants or this pollution have to be taken care of in a scientific manner. So this pyrene is a polycyclic aromatic hydrocarbon, and our project is on the biological remediation. This remediation is getting rid of these pollutants from our environment. So we almost there are very prominent physical, chemical and other approaches, but biological approaches are very benign. They do not harm the environment in a very detrimental way. So that is why we have taken up the biological remediation. Dr. Sunil will tell more detail about the biological remediation, but this is the requirement that we are working into. So in this project we have taken polycyclic aromatic hydrocarbons because they are very recalcitrant and very stable compounds which are very ubiquitous, and these compounds are they were degraded by using the fungus. So these fungus were isolated from in-house sources, and we use the fungus, and in the larger picture we are using not only fungus, we are using bacteria, the enzymes in different forms, and then causing these pollutants to degrade into the lesser toxic or non-toxic products. So the pyrene is a class of belongs to the class of hydrocarbons, although it is not toxic, but it represents the toxic forms of hydrocarbons. So that is why we have selected pyrene, because it is easier to handle this compound in the laboratory and without getting exposed into with this harmful chemical. So more details, Dr. Sunil.
गुड मॉर्निंग. जैसा कि डॉक्टर पंकज ने बताया कि रेमेडिएशन, आपने पूछा कि रेमेडिएशन को एलेबोरेट कीजिए कि क्या है इस वर्ड को. तो अगर आप रेमेडिएशन का शाब्दिक अर्थ देखेंगे डिक्शनरी में तो उसका मतलब होता है उपचार. तो उपचार किस चीज़ का करते हैं? जब कोई चीज़ बीमार हो जाता है तो उपचार करते हैं, इसका मतलब तुम यही समझते हैं. उसी तरह हमारे जो एंथ्रोपोजेनिक एक्टिविटी है उसकी वजह से हमारा इन्वायरमेंट हर दिन बीमार हो रहा है, वो हमारी एक्टिविटी की वजह से. लाइक डॉक्टर पंकज मेंशन दैट बाय ग्रोइंग द पॉपुलेशन, इंडस्ट्रियलाइजेशन, तो हर रोज़ नया नया चीज़ आ रहा है और उसमें क्या हो रहा है, एनर्जी का यूज हो रहा है. तो एनर्जी का मेजर सोर्स अभी भी 85% ऑफ एनर्जी का जो सप्लाई है वो क्रूड ऑयल और फॉसिल फ्यूल से होता है. तो जब फॉसिल फ्यूल को हम यूज़ करते हैं, बर्न करते हैं, तो उससे पॉल्यूटेंट्स जनरेट होते हैं. नॉट ओनली फ्रॉम द फॉसिल फ्यूल्स, देयर आर अदर सोर्सेस आल्सो लाइक बर्निंग ऑफ एग्रीकल्चर रेसिड्यू, बर्निंग ऑफ कोल. These are the forest fire. These are the major source for generating the pollutant like pH. तो उस pH जब उत्सर्जित होता है इन्वायरमेंट में तो वो आपके उसमें आता है, इन्वायरमेंटल मैट्रिक्स में आता है. मतलब एयर में आएगा, सॉइल में आएगा, वाटर में आएगा, और वो उसको आपके इन्वायरमेंट को पोल्यूट करेगा, आपके इन्वायरमेंट को वो बीमार करेगा. तो हमारा पर्पस ये है कि पेट्रोलियम रिलेटेड जो सोर्सेज से पीएच हमारे इन्वायरमेंट में आ रहा है उसको टैकल करना, और उसके लिए एक ऐसा बायोलॉजिकल टेक्निक जो कि सस्टेनेबल हो उसको यूज करके उसका उपचार करना. तो बेसिकली वी आर ट्रीटिंग दीज पोल्यूटेंट यूजिंग बायोलॉजिकल मेथड्स. So in this biological method we are using microbes, mainly fungus and bacteria, and as you mentioned that we have used Trematis maxima. So Trematis maxima is class of fungi, but apart from Trematis maxima we are using bacteria like Pseudomonas, Rhodococcus. So and we are preparing consortia of the microbes so that they can work together and enhance the capacity of degradation, because the natural wild type microbes have their own capacity. They cannot degrade on on the pace of your requirement. They will work on their own pace. So we provide the certain condition where they can work properly and degrade the pollutant faster than their actual capacity. So far, as mentioned, Dr. Pankaj has mentioned that we are degrading pyrene, but apart from the pyrene, we are taking more aromatic hydrocarbons, toxic compounds, because these toxic compounds are in priority list of US EPA. They have categorized 16 priority pollutants, and in that 16, Pyrene is one of them. And apart from the Pyrene, we are working on anthracene, chrysene, naphthalene, these pollutants. And so far, we have achieved the degradation of these compounds up to 4 ring compound in laboratory. And we are also testing this degradation potential of the microbes, which we have developed in our laboratory in real conditions. So we have tested our microbes in the soil also, and it is giving good remediation result, and that result is we are going to publish in such papers that will come, and we will share that result with you so that we can share this data to the person they are not in scientific field, but for the non-scientific person also, that they can read and understand what is happening due to the anthropogenic activity using the petroleum as in as.
Yes.
I think, as I mentioned earlier as well, the one factor that stands out here is accountability. Like the way you're working, like sitting at the Indian Institute of Petroleum and solving the pollutants and everything that comes out of it. I think that's again amazing. But I also want to understand from you that how can this be done at scale? Like will you be applying it at places, for example, when oil spills happen, or like how are we going to implement it at scale?
All right. I'll take that question. We know that this is a very important area, and a lot of researchers across the globe have been working in this area. The common practice is to degrade the petroleum hydrocarbons falling into different types of hydrocarbon classes. When I say hydrocarbon classes, they are either saturated hydrocarbons or unsaturated, olefinic, aromatics. They are sulfur containing compounds, nitrogen containing hydrocarbons. So there is a huge variety into the petroleum hydrocarbons. So most of the researchers have already quantified the degradation or the biodegradation potential of different type of microbes with respect to these hydrocarbon classes. But specifically when I speak about polycyclic aromatic hydrocarbons, they have been rarely studied because these compounds are very stable, very toxic, and very their structure is very stable. They cannot be degraded. They are persistent organic pollutants. They remain in the environment for a very longer time, and the natural processes do not facilitate their degradation at a very reasonable pace. So that is why this whole issue becomes very important, that not only the other hydrocarbons but the polycyclic aromatic hydrocarbons must also be included into the total petroleum hydrocarbons. And when we because this is an evolving phase, we see a lot of research papers coming into the literature, but all this has to mature into a product. So our target is to develop a very stable microbial consortium comprising of bacteria, funguses, and all other things, and then that consortium can be sustained in a very a way that in which it can be handled easily into, as you mentioned, oil spills. Oil spill has been historically a very grave problem which is prevalent across the world. Our country has also witnessed the Mumbai oil spill, Chennai oil spill. So the oil spill leads to very significant damage to the environment. So when it comes to the coast, when it comes to the soil, our target is to prepare a material that contains the microbial consortium immobilized onto it, and then that material can be used onto the soil or the sand or the polluted area, where that consortium can assist into easier or faster degradation of those compounds. So that societal concern is always there. We are working towards developing a consortium that is very robust and can not only degrade simpler hydrocarbons but polycyclic aromatic hydrocarbons. So that is our motto.
And for example, soil, water, it's everywhere, and I would like to believe that most of the areas it's polluted. So how many of these devices or these consortiums would be needed to solve this problem at a larger scale?
Yes, actually the consortium development does involve a huge collection of diverse type of samples. Like if we are a petroleum laboratory, we will emphasize only on the petroleum spills, either the terrestrial spill or the marine spill or any kind of other spills which is accidental. So when you say that everywhere it is polluted, so we would say that where there is a requirement, where there is urgency, there it can be used at priority. Otherwise, these microbes can be, microbes can be used in different forms to all the areas where it is polluted. But our concern is to provide this material to a high priority polluted area.
And we do not have anything of such kind like at the moment.
We have some products, but still the validity needs to be ensured for wider application. So we are just improving if anything is available. Of course, it's available globally, but we are on to improving their efficiency and make a robust this consortium.
So my last question to you would be that since you're using, fungus and, like using these natural, things that exist in the environment, like natural beings that exist in the environment, do you believe that that's a way we can actually solve pollution or tackle climate action in a way?
Dr. Sunil, please.
Yeah, actually, in environment there are microbes and fungus are already present, and they are doing their work for remediation. But as I mentioned, that their pace of working is like for their survival only. They are not, working for us. They are working for themselves. So there are microbes, but the thing is that we have to identify such microbes and the more potent microbes that can degrade the pollutant in faster way. So that's why in the microbiologists are searching lot of microbes from different geographical area. The meaning is that the geographical area is very important, because the microbes which is working here that may not work on another site, because their growing condition is different, because their survival condition is different. So it is very important for microbiologists to select a vast range of microbes and make a consortia for treating these pollutants. So this is in this direction we are also working. Apart from this polyaromatic hydrocarbons, we are targeting the pollutants which are coming from fertilizers, the pollutants which are coming from the apart from the petrochemical industry. We are also working on that, but that is on initial stage. Our students are working on that.
I'd also like Sunil to mention about the secretomic part, because enzymatic and we have already identified some enzymes. So if you'd like to mention the secretomic part.
Yes, because the microbes for degrading any pollutant or any compound, biotic compound, they use their machinery. And what is the machinery? Machinery is their enzymes. They produce a lot of enzymes inside that, and they take the pollutants and then they break into smaller compounds through various metabolic pathway. So we prepare the consortia and check in presence of the xenobiotic compound what that microbes are producing in that environment. Because suppose we are trying to degrade X compound, and the microbes doesn't have the potential to degrade X, so there is no use of that microbes in that consortia. So before doing any experimental plan for further broad study or in real-time study, we are studying the secretomic pattern of that microbes, how that microbes are behaving in presence of certain pH or any pollutant. Is that system available with that or not? So that is also we are taking care of that secretomic study. So how the pollutants are affecting their metabolic pathway, or the gene, how the gene is regulating, it is up-regulating or down-regulating, and what kind of secretory protein they are releasing in the environment. So this kind of a study we are performing right now.
And what would be like the kind of collaborations or things needed for you to actually take what you've developed to, for example, actually like into the oil spills or the marine spills that we call about?
First of all, we need access to the polluted matrix. The real world pollution, as you said, into the marine spills is accessible. And to develop a consortium beforehand, so we need to have an access to the sites where the spills is actually taking place. For example, if a pipeline is going from a refinery to a certain distribution terminal, so there is a leakage and there is a terrestrial spill, we need an access to that microbial consortium or the microbes present in that environment. Then when a crude is imported, it goes into the refinery. When during its handling it is spilled into the soil, we need access to those matrices. So when it is distributed at a retail outlet, it is again spilled on an ongoing basis. So accessibility of those areas is the first thing. If that accessibility is easy, I must say it is right now not easy to go and find a polluted site, and that too with an access to handle it, to see it and to collect it. So that is easier than that formation part of the scientific study planning is more easier.
Adding to this, on collaboration side, apart from this thing, I mean, knowledge is also required from the different areas. Suppose this is not just microbiology, and it requires a lot of bioinformatics things. It requires a lot of analysis, chemical analysis. Like that, Dr. Pankaj is chemical analyst, and myself is biotechnologist. But the interdisciplinary knowledge is very much required, because the application of biotechnology in environment is difficult. I mean, not easier than other field. Other fields have already, developed a lot of things. In agriculture, biotechnology application is there. In pharmaceutical, lot of biotechnology application is there. But in environmental field, it is still growing. So we required a lot of knowledge from the different area. This collaboration of the knowledge is also very much important.
I think it's very important. And just to finish it off, I myself have a background in medical biotechnology. I did my graduation in medical biotechnology, and one thing which I realized that after so many years also, and there are very few of my batchmates or seniors who have actually gone ahead and pursued a career in biotechnology. So one thing which I always get to hear is that what's the scope for me, what can I do, is it just a very long cycle? And so I want to ask you this question, because you are working in this field, that what are the opportunities for, say, students entering into the biotechnology field, and what should they be being focused on right now?
I think a biotechnologist would be more suitable for this question, but there are a lot of prospects available. And as Dr. Sunil mentioned, that it's an interdiscipline, cross-disciplinary area. Not a biotechnology person alone can handle this situation. We need to collaborate with material scientists, we need to collaborate with analytical scientists and microbiologists. So many researchers need to come at a common platform, and then we also need to have leaders who can conceptualize the problem. And then on the basis of empirical information available, we can give an hypothesis. So there is a lot of opportunity. If you like to add something for biotechnology especially.
Yeah, actually opportunity is there, but first of all we have to make up your mind that are you satisfied in this job? Is this job is actually for you or not? Because in our group also, in BTech and MTech, most of the students have they are pursuing their career apart from the biotechnology. They are working in KPOs, LPOs, and they are not working in this biotechnology area. As I mentioned, that the interest is more important. If you are not interested in research, that is not This will not help, because a lot of time is required, a lot of study is required. Every day you have to read what is going on in the entire world, in scientific community, what they are doing, where you are lacking. So these kind of thing is required. So if you are very much interested in research, definitely you can come and join the biotechnology in any field. Not in environment field, any field. There is a lot of scope in biotechnology. You do your PhD, do your postdoc, and join. In India still, when the current the leadership is focusing on the Atmanirbhar Bharat. So this is a good opportunity for the youngster who is doing BTech and MTech, do their PhD from CSIR, like our organization, and from apart also. They can go abroad and do their PhD and come and join the organization. There's a lot of fellowship available and a lot of opportunities there in India for biotechnologists. So I can say that this is a good field to start the career who is interested in research field.
Thank you for sharing that with us, and thank you for doing what you do. Thank you so much for being with us today.
Thank you very much for your time.
Thank you.